Hello ladies and gentlemen, and welcome to lecture 4. We're going to talk about ion channels and transporters here. So we'll talk about single channel and synaptic currents in the first part. Then we'll talk about the structures of a few different channels, and finally we'll wrap up by talking about the sodium-potassium ADPAs. This is essentially the battery that keeps this entire electrical system functioning. So synaptic currents are very important to us, but they arise from individual ion channels activating synchronously and then summing together. A synaptic current can actually have two components. We're going to think of them as just kind of one thing, just a synaptic current, but there are many things going on, and so sometimes you'll see multiple components where there's an early and a late part, and this can arise from ionotropic receptors and metabotropic receptors. Ionotropic receptors are going to activate rapidly and produce fast ion currents, whereas metabotropic receptors are going to activate later. And we can see that difference in timing here in this illustration. So on the left part in panel A, we can see the change in membrane potential that occurs following synaptic stimulation. You can see the stimulation artifact at the leftmost portion, that kind of spike that shows up. And depending on the membrane potential, you'll either get an inward or an outward current. The IV relationship is shown on the right there, so you can see the current or the membrane potential that results from the current at different membrane potentials. So you can essentially see where it reverses. So the early component reverses near 80, somewhere around 80, maybe 78, I guess the left illustration shows. So it's probably a chloride conductance. And then the later component, you can see, has a reversal potential near 100, so it's probably potassium. So you have an early ionotropic chloride component and then a late metabotropic potassium component. A lot of what we're talking about is going to be the ionotropic fast component. Okay, so the ion channels that are opening and closing here, they're doing so in a probabilistic fashion, but it's also digital. Okay, probabilistic meaning that just because the ligand binds, it doesn't mean that the ion channel will open. And just because the ligand isn't bound doesn't mean that the channel won't open. It's all operating based on probability. For example, when acetylcholine is puffed out onto this little tiny acetylcholine receptor there, here we can see it's an outside-out patch. That acetylcholine will bind to the ligand binding site, and what it does is increase the probability of channel opening. So if you look at the currents on the bottom of this illustration, you'll see it's cycling from open and closed back and forth, but there's never an intermediate, and that's what makes it digital. Each time that ion channel opens, it's going to produce the same reliable current for as long as it's open. And you'll notice that it's open for different periods of time, and that again goes back to the probabilistic component of this. So the ion channels are actually opening and closing the whole time, whether the ligand is bound or not. It tends to spend the majority of its time closed when the ligand isn't bound, and it tends to spend a whole lot more time open when the ligand is bound, but it's all a probability. Whether it's ligand or voltage gating, it doesn't matter. It's still a probability. So what we're looking at here would be the activity of some different voltage gated calcium channels. The very top part is showing you the current step that they deliver, since it's voltage gated. So right in the middle chunk of the recording windows in panels A and B, that's where we're depolarized. You'll see some channels open for a little bit, some channels never open, some channels are open for a long time, and some very briefly. So it's all just a probability. Alright, that's it. Now, of course, we can be a little more mathematical about this. When we're at the synapse, we can calculate the single channel currents using, of course, Ohm's law. That's all we're looking at. This is V equals IR. It's just rearranged a little bit. Instead of resistance, we're dealing with conductance. Remember that G is the inverse of resistance. So instead of V equals IR, it's I equals VG. So I is the current, G would be the conductance. Different channels are going to have different conductance. Some will have greater conductance depending on their open time, open probability. And then V, in this case, is the driving force. It's the difference between the membrane potential and the reversal potential for whatever ion we're talking about. But that's just Ohm's law, V equals IR. The single channel currents are all digital. It's all or none. It's open or it's closed. The synaptic currents, though, those are going to have shades of gray depending on how many receptors are going to be open. So you can see the equation here for the synaptic currents is very, very similar to the single channel currents. There are just two additional variables. There's the open probability of the channel, and there's the number of receptors or channels at the synapse. A synapse with more channels has the potential to have a greater synaptic current. A synapse with channels that have higher open probability will have greater synaptic currents. So all those individual channels are essentially being summed together, and we estimate that with that equation there. You won't need to know this. This is just for the mathematically minded folks. But if you look on the right, you'll see on top, those are the single channel currents. Some are open for a brief period of time, some for longer. And when you kind of add all of those up, at the very beginning, all 5 are open. Very soon after, one of them closes, a little bit later another closes, then another, and then a little bit later the last one closes. So you get this very rapid maximal response and then a slow grading back down to nothing. And that's what you see at the synapse. That's what's shown in part B. You get an early strong activation, and then the number of channels that are open starts to decrease over time. And that's what gives you the smooth synaptic currents that we see. If the current is excitatory, that's going to depolarize the cell. And this is going to be caused by the inward movement of cations, of positively charged ions. Some channels will be sodium channels, so they'll be specific for sodium. They're going to reverse at 56 millivolts. Others will be non-specific cation channels. They'll let sodium come in, they'll let potassium leave, they'll let calcium come in. They don't really care. They just let cations move. That will reverse around 0 millivolts. This is still more than sufficient to drive an action potential. And that's the goal of excitatory currents, to drive an action potential. Okay, depending on the strength of the current, we may or may not get an action potential. Okay, if we get above threshold, we'll see that we drive an action potential. That's what we're seeing on the bottom there. These can propagate to the dendrites, but really all we care about is that black label on the bottom showing us the action potential being driven with the strong excitatory input. So what they're just showing you is there's a few black lines where we don't get an action potential, and that's simply because the excitation wasn't strong enough. On the other hand, inhibitory currents are there to prevent action potentials from firing. In some cases, the inhibitory currents will hyperpolarize the membrane potential. But for the most part, what's happening is what we call shunting inhibition. And that's where the inhibitory currents just simply prevent depolarization rather than causing a strong hyperpolarization. For example, if you open a chloride channel, the reversal is very close to rest, and so the chloride current will be minimal until the neuron starts to depolarize. So if the chloride channels are open and you receive excitatory input, think about what that does to the driving force. Okay? If the reversal for chloride is minus 76 and we're resting at minus 70, that's a very small 6 millivolt driving force. But let's say we start to depolarize and we become more positive. What does that do to the driving force for chloride? Well, we're moving further away from it, right? If we now compare negative 76 to 60 millivolts, well, now we have a 16 millivolt driving force. And so as the neuron depolarizes, you get a greater driving force for chloride, and so this actually resists that depolarization. That's mostly what inhibitory input's about. There's some hyperpolarization that occurs, but that's largely reserved for potassium conductances. Chloride is mostly about shunting inhibition. So you can see shunting inhibition in effect on this illustration. If you look in the top part, you'll see control is showing you just regular old excitatory input. We haven't activated any inhibitory input. The green trace is showing you the same excitatory input, but now with a chloride conductance. And what you see is that that excitatory input has been shunted. It's been reduced. And that's really what inhibitory input does. Don't think about it as hyperpolarization. This is where most folks start to think about it, but it's a little inaccurate. Mainly what inhibition does is reduce excitation. And as a result, you can see in the bottom panel, the firing rate of a neuron in response to excitatory input becomes much lower when that inhibition has been activated. And so we have to have fairly high excitatory input in order for us to start firing action potentials. So at the point where it becomes non-zero in this model, well, we're already firing at around 200 hertz. So inhibitory input is there not so much to hyperpolarize, but instead to resist depolarization. And that's what shunting inhibition is. So we get our excitatory or our inhibitory input in the dendrites, and then the cell body puts it all together and determines whether or not we fire an action potential. Now you might think that if you have 10 millivolts of depolarization in one dendrite and 10 millivolts in another, that's going to lead to 20 in the cell body. Not at all true. Of course it's not true. First of all, V equals IR, and the resistance is very different in the dendrites than the cell body. But even if that weren't the case, there's still going to be differences in how these currents sum. Part of it has to do with the change in driving force. Let's not forget driving force. So let's say that you open up a nonspecific cation channel. This reverses at 0 millivolts. This causes some depolarization. What happens to the driving force as the neuron depolarizes? So it's sitting at minus 70 at rest. If reversal is at 0, we have a driving force of 70 millivolts. If you depolarize and now you're at minus 60, well, now you only have a 60 millivolt driving force. And so if the neuron is depolarized when it receives another excitatory input, it's not going to get the same amount of depolarization. Okay, we can see that in this panel. If we look at part B there, what it's showing is the summation of the same synapse over time. So you can see when synapse 1 is activated, the motor neuron that they're recording from gets a little bit of depolarization. If you were to stimulate it twice, you get a little bit of depolarization, and then a little bit more that's not quite as much as the first one. Stimulating it twice doesn't give you double the depolarization, and that's because of the change in driving force. You see this whether you sum over time or space. If you were to activate 2 excitatory synapses at the same time, you won't get double the excitation. You'll get something very close, but not quite the double. Now, in addition to this, there are also some channels that will only open up when the neuron is depolarized a little bit. Some ligand-gated channels are also, to some degree, voltage-gated. The NMDA receptors are the principal example of this, and we're going to talk about them in much more detail when we talk about learning. NMDA receptors are not active at rest because the negative membrane potential pulls this magnesium ion into the ion pore and blocks it. So only a depolarized neuron can have NMDA receptors get activated and produce those excitatory currents. So you actually might find that depolarization gives you a stronger excitatory input in some cases. Okay, let's talk ion channels. We're going to talk general structures and then how these are going to determine their properties. Okay, a couple things to keep in mind. Not every ion channel is going to have the same components. Okay, they're all going to have an ion pore because the lipid bilayer is going to be impermeable to ions. That hydrophobic core is going to prevent charged molecules from freely diffusing. That's why we need ion channels in the first place. So we have to have the ion pore. That's the very light blue component of this cartoon that I've drawn here. So this is just an opening in the membrane, essentially, that's lined by the protein and allows certain ions to pass through. Which ions pass through totally depends on the ion channel. And there's a component of it called the selectivity filter that determines what ions can pass. And it does so by size and charge. Then there are some optional components. Some ion channels require some stimulus to be activated. Okay, they could be activated by the membrane potential, like those voltage-gated ion channels, or they could be activated by a ligand, like neurotransmitter receptors. The activation gate is the portion of an ion channel that's required to open the pore. Not every channel needs these. The leak channels that establish resting membrane potential don't have an activation gate. They also lack an inactivation gate. Okay, this is the portion of the ion channel that actually blocks the pore and turns it off after it's turned on. Not every channel has this. Voltage-gated sodium channels have it, but voltage-gated potassium channels typically don't. Okay, so those last two are optional, but they will allow the ion channels to behave in some pretty cool and interesting ways. Okay, there's a lot of different ion channels out there, and they're going to differ in a few different respects. First of all, ion selectivity. They have different selectivity filters. They'll let different ions through. There can be some ion channels that allow any cation through, and then there will be others that only allow sodium to move through. It totally depends on the channel. Others differ in terms of their gating. Okay, ligand versus voltage gating. If there even is gating, right? Remember, not every channel has gating. Leak channels have no gating. They're always open. Ligand-gated ion channels need a ligand to bind in order to pull open the ion pore. Voltage-gated ion channels usually have some sort of a voltage sensor, positively charged amino acids that are repelled from the positive charge that enters. This rearranges the ion pore and allows ions to permeate. And again, some are inactivating, some aren't. Voltage-gated sodium channels should inactivate. If there's a mutation, then they can lack this inactivation gate, and they don't fully inactivate. Other channels just don't even have the inactivation gate, like your typical voltage-gated potassium channel. Then there's the kinetics, the rate at which they open and close. Voltage-gated sodium channels have faster kinetics. They tend to open sooner rather than later, whereas the potassium channels tend to open later than voltage-gated sodium channels. The ion pore opening varies, so does the ligand binding kinetics. Some receptors will bind their ligand very quickly and very tightly. Others will bind it very slowly. Some allow rapid binding and unbinding, whereas others allow very slow unbinding after the ligand binds. And that will, of course, affect the probability with which the ion channel is opening. So there's a lot of diversity out there, and the important distinctions will be highlighted later on. Then last but of course not least, they differ with respect to pharmacology, what drugs act on them. So those NMDA receptors that we will discuss later, there are many different subunits that make them up, and depending on the subunits that they comprise, that'll determine what drugs can bind to them. Okay, so different receptors are going to be activated by different drugs, and even different types of the same receptor can be activated by different drugs. We're not going to go over all the ion channels under the sun. That'd be ridiculous. That'd take several classes, and I mean full semesters, to actually cover them. Just keep in mind there's a lot of diversity, but it all comes down to these four different things. What ions move through, what's the gating, what are the kinetics, and what are the drugs that can act on it. Okay, now let's talk about the things that really matter to us. Let's revisit the action potential. That voltage sensor, that activation gate, okay, that's all brought about by charged amino acids. That's it. It's not magic. The voltage-gated sodium and potassium channels have positively charged amino acids in the fourth transmembrane domain of each subunit. that's going to interact with positive charge that enters the cell. So when the membrane potential depolarizes, that positive charge is going to interact with those positively charged amino acids and cause them to move. They kind of spin a bit in voltage-gated sodium channels. They tilt over in voltage-gated potassium channels. Doesn't matter. In the end, they move, and this opens the ion pore. Now, voltage-gated sodium and potassium channels also differ in terms of kinetics and gating, as well as drugs that act on them. But here we'll talk about kinetics and gating. The voltage-gated sodium channel is going to activate very rapidly. Okay, so immediately after the depolarization, that's what we see on the top of this. All right, the top is showing you membrane potential over time. So that little square wave that steps up, that's showing you very substantial depolarization. When that occurs, we are well above threshold. Notice the voltage-gated sodium channel is going to open before the voltage-gated potassium channel, so they differ in terms of their kinetics. That's very important. That allows that initial rapid depolarization. If the potassium channel opened at the same time, the potassium current would offset the sodium current. And instead of getting strong depolarization, you get incredibly weak depolarization. They also differ in terms of gating. The voltage-gated sodium channel inactivates to create that absolute refractory period where you cannot fire another action potential. Voltage-gated potassium channels typically don't do that. Some do, but most don't. And that allows them to stay open for longer and cause that repolarization that occurs in the action potential. So you see two key distinctions here. Faster kinetics in sodium channels and inactivation in the sodium channels. Now you'll notice I said potassium channels usually don't inactivate. Look, there's a ton of different potassium channels out there. This is a very diverse class of proteins. For example, there are some leak channels. A lot of the resting membrane potential, a lot of the leak is because of potassium. So some are leak channels, some are voltage-gated potassium channels. Others might be gated by ligands like ATP. And then inactivation can actually occur. Kv 2.1 channels don't inactivate, but there are others who do. Kv 4.1 is a different potassium channel that does rapidly inactivate. And then there are other potassium channels that inactivate so rapidly that they actually will only conduct after repolarization has occurred. So potassium channels are highly variable. I do want you to know about Kv 2.1 because those are your prototypical potassium channels. Okay, they're voltage-gated, non-inactivating potassium channels. Those are the ones that we think about whenever we're talking about your prototypical voltage-gated potassium channels in the action potential. Now, of course, there's ligand gating. Okay, the activation gate can be triggered by the binding of ligands. And that's all neurotransmitters are. They're just little chemicals that come and bind to receptors. And when they do that, that essentially pulls open the ion pore. Now remember, it's probabilistic, okay? It's not that they all open simultaneously and stay open as long as the ligand's there. When the ligand's bound, it's more likely that the ion pore will be open, and then all of the individual ion channels will sum to create the synaptic currents that we see over there. Now, just like the voltage-gated channels that we've talked about, it's still the ions are going to move according to their electrochemical gradients. Okay, so they're going to move by diffusion, but they will also move according to membrane potential. Okay, positive charge will still repel positive charge, and they'll still move from high to low concentrations. Okay, so the reversal potential that we've discussed before still holds true even with ligand-gated ion channels. Same idea here. Potassium channels still reverse at about -100. Non-specific cation channels reverse at 0. Sodium channels reverse at about +56. Some activation gates are actually mechanical. So we have what we call stretch receptors that are exactly what they sound like. They detect stretch. They're physically pulled open. We use these for somatosensation, so we feel around the world using stretch receptors when we touch something that causes our skin to dent inward. This stretches membranes and actually pulls open the stretch receptors. That forces open the ion channel and ions move through the membrane. There are other channels that are going to be activated by environmental stimuli, like temperature, pH, some intracellular molecules that should stay inside of cells, but if they get killed, then they'll be outside. So these are typically going to be used for nociception or detecting pain. And the channels that are contained on these nociceptors are called TRP channels, or transient receptor potential channels. Some are activated by heat, some by cold, some by acid. This is a varying family of molecules. But what they're detecting isn't really a ligand per se. I guess pH kind of is. Instead, what they're detecting is the environment. What's the temperature? Is it really hot? Is it really cold? And then there's the pharmacology. Different receptors are going to be acted on by different drugs. For example, the voltage-gated sodium channels that allow the action potential to take place are inhibited by tetrodotoxin. This is a toxin in pufferfish, and by preventing the activation of voltage-gated sodium channels, tetrodotoxin can be incredibly lethal. There are other toxins that actually prevent inactivation rather than activation. Those would be the alpha toxins from scorpions. So you'll get more prolonged sodium currents with these toxins. Then there are beta toxins that facilitate activation at lower membrane potentials. Okay. Both of these are kind of doing similar things. They're going to be toxic because they're increasing the excitation of neurons to a point where it starts to kill them. So without the inactivation of sodium channels, or by facilitating their activation when it shouldn't occur, this leads to hyperexcitation and eventually neuronal death. These can be nasty things. The potassium channels are also activated by a number of different toxins. There's a tetraethylammonium, which can block the voltage-gated potassium channels, but won't block voltage-gated sodium channels. So again, there's going to be differences in the pharmacology for different ion channels. So far, what we've talked about is passive transport. We got a bunch of sodium outside, we got a bunch of potassium inside. When we open a sodium channel, sodium flows in. When we open a potassium channel, sodium flows out. This is all passive. But this will eventually run out if we never recharge our ionic imbalance. And that job is handled by pumps. The most important pump would be the sodium-potassium ATPase. This is going to burn one ATP molecule to export 3 sodium and bring in 2 potassium. Alright, we're going to talk about energy. It's important to realize that our brain uses a lot of energy. Alright, when we compare the mass of our brain to the rest of our body, we see it's only about 2% of our total body mass. But it's going to use about 20% of our oxygen and about 25% of our glucose, with glucose being the primary energy source for the brain. So you can see it's on a ratiometric level here, using about 10 times what you'd expect in terms of oxygen and glucose. And that is because neurons themselves use a lot of energy. This is because they have very high surface area to volume ratios. If you compare the surface area to volume ratio of a skin cell with a motor neuron, you can see that they are not even in the same ballpark. Okay, neurons have these very intricate structures. They have those branching dendrites and axons, and they're very thin. That creates a very high surface area to volume ratio. You should keep in mind that that surface area, that's littered with proteins. It's littered with ion channels that are moving ions all the time. That's what's accumulating a lot of debt here. So if we break down the energy usage for a neuron, we can see that they're using energy in a lot of different places. What we've talked about so far, the resting potential and the action potential, that's only about 30% of what it does. Then there's gene expression, that'd be the housekeeping, postsynaptic receptors, presynaptic release, and the recycling of neurotransmitters accounting for the rest. Okay, so the neuron has a high energy budget, and actually a lot of its energy is spent on maintaining that ionic imbalance. So around 25% of the ATP used by a neuron is used to fuel the sodium-potassium ATPase. Okay, because the ionic imbalance is constantly being perturbed by leak channels at rest and by gated channels during synaptic transmission or the firing of action potentials. Anytime you open an ion channel, the ion's going to move down its concentration gradient until it reaches electrochemical equilibrium, until it reaches reversal potential. Because we're moving ions around, we're disrupting that ionic imbalance, and so we have to actively maintain it. That's carried out by a number of different transporters. There's, of course, transporters to move around calcium, but the one that I want to talk about is the sodium-potassium ATPase, because this uses so much ATP. What it's going to do is remove 3 sodium, and it's going to bring in 2 potassium, all at the extent of 1 ATP molecule. Okay, so here it is in action. The inside of the cell is on the bottom, the outside is on top. We're going to take 3 sodium ions from the inside and move it to the outside. We're going to do that by burning ATP. All right, there's ATP, adenosine triphosphate. Those triphosphates have high energy bonds that are broken readily, and the free energy from them are used to drive some sort of cellular process. It could be the making of bonds, the breaking of bonds, or just simply the rearrangement of a protein like the sodium-potassium ATPase. Okay, so we're moving sodium against its concentration gradient. We do that by only exposing sodium binding sites toward the inside of the cell. Now that ATP and sodium have all bound, the protein hydrolyzes ATP. That phosphate group stays attached. Its negative charge causes the protein to rearrange. It destabilizes the sodium binding site. Sodium spits out to the outside of the cell, and then it opens up the potassium binding sites. After potassium binds, this destabilizes the interaction with the phosphate. The phosphate group falls off. Protein rearranges. The potassium binding sites become destabilized. Potassium leaves, and now we have our sodium binding sites. Open again. Sodium can bind. Again, ATP will bind. There will be ATP hydrolysis where we break it. That phosphate group stays. The protein rearranges. Sodium leaves. Potassium will bind and enter. Rinse and repeat. This is cool stuff. That ion transport not only recreates the ionic imbalance that we need to carry out the action potential and all those synaptic currents that we've discussed, it's also electrogenic in its own right. In other words, it creates an electrical current. We're moving out 3 positive charge, but only bringing 2 positive charge back in. That means we've generated a net -1 charge. So over the long run, the sodium-potassium ATPase helps maintain resting membrane potential by creating not only the ionic imbalance needed for the leak channels to operate properly, but also by generating negative charge itself. Now, if we were to look at the structure of the sodium-potassium ATPase, you'll see that there's an alpha and a beta subunit. In the alpha subunit, we have a Wabane binding site. That's the inhibitor, which we'll see in action in just a moment. There's the phosphorylation site, which gets phosphorylated after ATP binds to allow the rearrangement. And then we have some negatively charged amino acids that will bind sodium and potassium. If we were to inhibit the sodium-potassium ATPase, we can see how important it is. So in panel A, we're not doing anything at all to it, and we can see that this neuron is resting, let's see here, somewhere in the ballpark of -80 millivolts. Whenever we add the inhibitor Wabane, at 10 micromolar, 100 micromolar, or 1 millimolar, we see that in all cases we get some depolarization. And that's because we're no longer exporting that sodium that comes in and bringing back in the potassium that leaks out. Both of their reversal potentials will eventually run to zero, and thus we're going to depolarize. We're also not getting that -1 charge out of the electrogenic transport from the sodium-potassium ATPase. So in panel B, we can see that by adding higher concentrations of Wabane, we get more and more depolarization. Now, of course, there are other pumps that help offset the ionic imbalance. That help create the ionic imbalance. And if we further inhibit them with iodoacetate or sodium cyanide, we can see that we get a little bit more depolarization. But without this active maintenance of our ionic imbalance, you can see that the membrane potential falls apart. We don't rest at negative values without our pumps operating. All right, so those are the batteries that keep the membrane potential at the appropriate charge. Now, because these pumps have to operate, that means that the neuron requires ATP. And in fact, every time it fires an action potential, it uses about 1 second's worth of ATP that it could have used to maintain resting membrane potential. So in the span of 3 to 4 milliseconds, it's burning up 1 second of energy. So action potentials are fairly costly. They're about 250 times more expensive than just rest. That means if we're going to fire an action potential, we need to make sure we have enough ATP around. The neuron has ATP-sensitive potassium channels to do just that job. So they sense ATP levels. They're blocked when ATP levels are high. So as long as we have enough ATP around to keep our pumps running, these potassium channels stay closed and we can fire action potentials. That's what we're seeing in panel A. All right, now remember, potassium channels are going to hyperpolarize the cell because potassium reverses at -100. So potassium channels are inhibitory. They hyperpolarize and they have the shunting effect. So when ATP levels are low, the KATP channels are no longer blocked. They open up. We increase potassium conductance. If you revisit the Goldman-Hodgkin-Katz equation, you'll see that if you increase the potassium permeability, you hyperpolarize the membrane potential, and as such, it becomes much more difficult to fire an action potential. So if you don't have enough ATP around to keep your pumps running, these KATP channels open up, the cell becomes hyperpolarized and shunted, and it doesn't fire action potentials. It doesn't burn up more ATP that it doesn't have anyway. It's already in metabolic debt, so it shouldn't fire action potentials and further stress itself. This is a beautiful illustration of homeostasis, and it's completely understandable if you understand reversal potentials and how they tie together in the Goldman-Hodgkin-Katz equation. All right, that about does it. Fairly brief lecture. Email me any questions you have on the material. I look forward to seeing you for Lecture 5.